So far we've ingested food, we've taken that food down into the stomach where we began more mechanical and chemical digestion. So before we move into the major part of digestion where most of our food is broken down and the nutrients are absorbed, let's take a quick overview of the activities and the overlapping phases of digestion. The cephalic phase of digestion can be initiated by the smell or the sight or the thought of food, and it activates centers in the brain, in the cerebral cortex, the hypothalamus, and the brainstem, which then are going to activate some of our cranial nerves. The cranial nerves are going to stimulate the salivary glands to secrete. The vagus nerve, cranial nerve number 10, is going to stimulate the gastric glands to secrete their juices. And this is all to prepare the mouth and the stomach for the food that we're about to eat. The gastric phase begins once food reaches the stomach. Neural and hormonal mechanisms regulate the gastric phase and promotes the secretion of gastric juices and gastric motility. The neural regulation occurs when food of any kind distends the stomach and stretch receptors in the walls, so those receptors are stimulated. Chemoreceptors in the stomach monitor the pH of the chyme. When the stomach walls are distended or pH increases because of proteins, and those proteins have buffered some of the stomach acid, the stretch receptors and the chemoreceptors are activated, and it sets up a negative feedback loop that you can see here on the right-hand side of the screen. The result of these nerve impulses is going to cause peristalsis and continue to stimulate the flow of the gastric juices from those gastric glands. Hormones also regulate the gastric phase. So gastric secretion during this phase is regulated by a hormone called gastrin. So gastrin is released from the G cells that we learned about a while ago in response to several different stimuli. It can be distension of the stomach by chyme, partially digested proteins in the chyme, high pH of chyme due to the presence of food in the stomach, caffeine, acetylcholine from parasympathetic neurons. Whatever the stimulus is, once it's released, gastrin enters the bloodstream because it's a hormone. It makes the whole round trip through the body and then reaches target organs in the digestive system. The gastrin is going to stimulate gastric glands to secrete a large amount of gastric juice. It also increases the contraction of the lower esophageal sphincter so that reflux doesn't occur. It increases the motility of the stomach and relaxes the pyloric sphincter, which helps to promote gastric emptying. Gastrin secretion can be inhibited when the pH of the gastric juice drops below 2.0, and it can be stimulated further when the pH rises. This negative feedback helps to provide the proper pH, low pH, for functioning of pepsin, for killing of microbes that may have entered the stomach, and for denaturing proteins of the stomach. The intestinal phase of digestion begins once the food enters the small intestine. These activities are also regulated by neural and hormonal mechanisms. The neural regulation is when nerve impulses are sent to the medulla oblongata due to the presence of the chyme in the duodenal wall. As a result of this, gastric motility is inhibited, and there's an increase in the contraction of the pyloric sphincter, so that increasing the contraction of the sphincter slows down the rate that chyme is emptied into the duodenum. The hormonal phase is mediated by two hormones that are secreted in the small intestine, cholecystokinin and secretin. Cholecystokinin is secreted by the intestinal glands in response to chyme containing amino acids from those partially digested proteins and also from fatty acids that resulted from partially digested triglycerides. It stimulates the stimulates the secretion of pancreatic juices that has a lot of digestive enzymes. It causes the contraction of the gallbladder so that the walls of the gallbladder squeeze and that stored bile is moved into the cystic duct through the common bile duct, and then it causes relaxation of the sphincter that allows that bile to flow into the duodenum. The cholecystokinin also slows gastric emptying by promoting contraction of the pyloric sphincter, helps to produce a full feeling so that it doesn't, so that we are prevented from wanting to eat more. And that's because it acts on the hypothalamus. It helps to also promote the normal growth and maintenance of the pancreas and enhances the release of secretin. Acidic chyme from the duodenum stimulates the secretin from the S cells that we saw a while ago in the intestinal glands in the small intestine. Secretin stimulates the flow of pancreatic juice that has a high content of bicarbonate ions. So these bicarb ions help to buffer that acidic chyme as it enters the duodenum. Secretin inhibits the release of gastric juices, also helps to promote the growth and maintenance of the pancreas, and enhances the act of cholecystokinin. Secretin causes a buffering in that chyme that reaches the duodenum and slows the production of stomach acid. Now, the importance of that, remember that your digestive enzymes are proteins, and when the secretin causes the pancreas to release those bicarb ions help to neutralize the acids in that chyme, because at those low acidic pHs, even the enzymes could be denatured, and that would slow the chemical digestion of the food in the duodenum. Once food moves into the duodenum, then we have to rely on the chemicals that are going to be secreted from the pancreas, the liver, and the gallbladder. So we'll start with the pancreas, which is a long, slender organ, about 5 to 6 inches long. It lies posterior to the stomach. It consists of a head, a body, and a tail. It's usually connected to the duodenum by 2 ducts. The head is the expanded portion that lies in the curve of the duodenum, the first curve. And then, of course, you have the body, which is the main part, and the long tapering tail. Pancreatic juices are secreted by exocrine cells into small ducts that unite to form the 2 larger ducts, which are the pancreatic duct and the accessory duct. These are going to secrete into the small intestine. The pancreatic duct is the large one that you see running the length of the pancreas. In most people, it joins the common bile duct of the liver and the gallbladder and enters the duodenum at a dilated common duct that's called the hepatopancreatic ampulla. An ampulla is simply an enlargement, and that's going to open into the duodenal mucosa. This is regulated by smooth muscle that surrounds the ampulla, and that muscle, surrounding muscle, is a sphincter that contracts and releases, and that is known as the hepatopancreatic sphincter. So the pancreas, if you look at it histologically, you've considered when we studied the endocrine system, and we saw that the biggest part of the pancreas was exocrine in its function, and perhaps you heard that you would learn more about that when you got to the digestive system, because only the small little patches of cells called the islets of Langerhans were producing those hormones. Remember the insulin and the glucagon and the somatostatin that were produced in the islets. Well, now we want to talk about the 99% of the rest of the pancreas, and those are the cells that are glandular epithelial cells that are in clusters called acini. So this is the exocrine part of the organ, and the cells within the acini secrete a mixture of fluids and digestive enzymes called typically pancreatic juice. The pancreas secretes somewhere around 1 to 1.5 quarts of pancreatic juice a day. Most of it's water, but it also contains some salts and sodium bicarbonate and several enzymes. The bicarbonate gives the pancreatic juice a slightly alkaline pH that buffers that acidic chyme. It stops the action of pepsin from the stomach and creates the correct pH for the action of the digestive enzymes in the small intestine. So the enzymes in the pancreatic juice include a starch digesting enzyme called pancreatic amylase, several enzymes that digest proteins into peptides, including trypsin, chymotrypsin, and carboxypeptidase, pancreatic lipase that's going to digest triglycerides, and nucleases such as ribonuclease and deoxyribonuclease that are going to break down nucleic acids and nucleic acids, RNA, and deoxyribonucleic acids, DNA, into nucleotides. Inflammation of the pancreas may occur with chronic alcohol abuse or chronic gallstones, and it's called pancreatitis. A more severe condition known as acute pancreatitis is associated with heavy alcohol intake or biliary tract obstruction. That's just blockage of some of the ducts associated with the secretion of bile. Patients that are suffering from this may release the active form of trypsin instead of the inactive trypsinogen, and this can begin to digest some of the pancreatic cells. Patients with acute pancreatitis usually respond well to treatment, but recurrent attacks tend to be what happens more often. In some people, though, the pancreatitis is idiopathic, which simply means the cause is unknown. Other causes of pancreatitis can include cystic fibrosis, high blood calcium levels, high levels of blood fats, some drugs, and some autoimmune conditions. However, in about 70 to 80% of adults with pancreatitis, the cause is alcoholism, and the first episode usually begins between the ages of 30 and 40. Pancreatic cancer usually affects people over the age of 50 and occurs more frequently in males than females. There are few signs until the disorder reaches a very advanced stage, and usually not until it's metastasized to other parts of the body, such as the lymph nodes, the liver, and perhaps the lungs. The disease is almost always fatal, and it's the fourth most common cause of death from cancer in the U.S. Pancreatic cancer has been linked to diets that are high in fatty foods, high alcohol consumption, genetic factors, smoking, and episodes of chronic pancreatitis. As we mentioned before beginning the pancreas, that chemical digestion in the small intestine is going to depend on the accessory organs, the pancreas, the liver, and the gallbladder. So we typically group them all together before we consider the small intestine and what's going on there. So since we just covered the pancreas, we're going to move now to the liver and the gallbladder, and I want to show you some images that give you the relationship of these three organs. So here, of course, you see in the cadaver, here is the duodenum, here is the pancreas, here is the liver, and here is the gallbladder. If you look at the diagram above, you can actually see the relationship and the ducts from the gallbladder, the liver, and the pancreas, and how they enter into the duodenum. So since we covered the pancreas, here's the large central duct, and you can see it comes in and enlarges at an area called the ampulla. The liver is going to make bile. That bile is then secreted into hepatic ducts, which have collected the bile from the areas within the liver and merged into your right and left hepatic duct. Your right and left hepatic duct then merge into a common hepatic duct, and then you have the common bile duct that comes down and meets the pancreatic duct here at the ampulla. Now, normally the hepatopancreatic sphincter has this duct closed so that the secretions from the pancreas and the gallbladder cannot get into the duodenum unless cholecystokinin has been released, and that, of course, is in the presence of food. So if there's no food in the duodenum, the hepatopancreatic sphincter is closed. Now let's think about what that means for the ductwork that is associated with the bile duct. So if the hepatopancreatic sphincter is closed here, then the bile that is being created out here in the liver is flowing into the hepatic ducts and then into the bile duct, but it can't get out because the sphincter is closed. So it backs up the common bile duct into the cystic duct. The cystic duct is the one attached to the gallbladder. So it fills the gallbladder, but it also is the one that empties the gallbladder when that cholecystokinin causes the smooth muscle in the gallbladder to squeeze, and then emptying the tube is the cystic duct, and then the bile flows down through the common bile duct to the hepatopancreatic sphincter that is now open because of the presence of cholecystokinin. And here you can see a larger version of the common bile duct coming in, merging with the pancreatic duct, the hepatopancreatic sphincter, that will keep them closed unless the cholecystokinin has triggered it to open. The liver is the heaviest gland in the body. It weighs about 3 pounds in an average adult. Of all the organs in the body, it's second only to the skin in size. It's inferior to the diaphragm and occupies most of the right hypochondriac and part of the epigastric regions of the abdomen. Now think about those words, hypochondriac. Hypo means under, and you should know that chondri refers to cartilage. So this is the area under the right ribs. Epigastric, above the stomach. So the liver occupies most of those two areas. The gallbladder is a small pear-shaped sac that's located posterior to the liver. So it's on that posterior surface. It's usually about 3 to 4 inches long and typically hangs from the anterior and inferior margin of the liver. So you can see it pokes out just a little bit. The liver is divided into 2 major lobes, the right and the left lobe, and also smaller quadrate and caudate lobes that you'll see when you learn the different areas of the liver in your lab. They're not visible here in this, but I often teach that the caudate is near the cava, near the vena cava, and the quadrate, quad means 4, it's a small 4-sided lobe on the inferior surface of the liver. Here you can see, as we talked about just a minute ago, that flow of bile as well as the pancreatic juices. So the right and the left hepatic ducts collect bile that is made out in the lobes of the liver and form the common hepatic duct where the right and left merge. That common hepatic duct then flows into the common bile duct, and if the sphincter is closed, the pancreatic juices are not being secreted, that bile then backs up into the common, backs up into the cystic duct, fills the gallbladder, and then it is emptied by the cystic duct when that gallbladder contracts. Histologically, the liver is made up of liver lobules, which are considered the functional unit. If you look deeper into those, you're going to see that there are hepatocytes, which have a number of functions, the Kupffer cells, which work as macrophages to clean up debris, old blood cells, bacteria. But also as a part of those lobules, you have the biocanaliculi, which are small ducts between the hepatocytes that collect bile that the hepatocytes are producing. Those small little biocanaliculi are going to merge into larger and larger and larger ducts that eventually fill the left and right hepatic ducts. So the canaliculi receive the bile that is made from the hepatocytes and then merge it into larger and larger ducts. Also, as a part of these lobules, you have hepatic sinusoids, which are capillary beds that are very, very highly permeable. And you find these between rows of hepatocytes, and these blood vessels receive oxygenated blood from the hepatic arteries and nutrient-rich deoxygenated blood that flows back in through the hepatic portal vein. Remember, that's coming in from the digestive organs. Now, the hepatocytes are going to be the ones producing the bile. They also convert nutrients, such as glucose, into the storage molecule, long-branched glycogen, helps to store vitamins, detoxify blood by converting ammonia to urea. And we'll look a little more closely at some of these functions when we move into a slide that's coming up in 1 or 2. All right, so, whoa, back up. So, I swear this touchy mouse, guys, I know y'all are tired of hearing it. I'm tired of dealing with the darn thing, too. So I just wanted to remind you about the Biorelease. Remember that hepatopancreatic sphincter is closed when we don't have that acidic chyme that is in the duodenum. That cholecystokinin that is released in response to that acidic chyme is going to cause the hepatopancreatic sphincter to relax and also stimulate the gallbladder to contract. That forces that bile through the common bile duct, and it is emptied into the lumen of the duodenum. It also is going to, the cholecystokinin is going to trigger the release of those pancreatic juices, including the bicarbs that neutralize those proteins. Bile emulsifies fats, which means it breaks big fats down into smaller fats. And again, we'll look at that when we look at the functions coming up. Those hepatocytes secrete about 1 quart of bile every day. Bile is a yellow, brown, or sort of olive green colored liquid with a pH that ranges between 7.6 and 8.6. So is that acidic or alkaline? Yeah, I know, that was an easy question. It's alkaline. Mostly water, bile salts, some cholesterol, phospholipids, bile pigments, and other ions make up bile. The principal bile pigment is bilirubin. If you remember when we talked about red blood cells and how they're broken down, that's going to create free iron and globin and bilirubin. The bilirubin is secreted into the bile and is eventually broken down in the intestine. Bile salts play a role in emulsification, and that simply means breaking down big lipids into smaller lipids. So it's not a chemical digestion. It is mechanically breaking down big fats into smaller fats. These small pieces allow for a very large surface area so that pancreatic lipase can work easier to accomplish the chemical digestion of triglycerides. The bile salts also aid in the absorption of the lipids after they've been digested. So let's do an overview of the functions of the liver and the gallbladder. So the liver is important in maintaining normal blood glucose levels. You should have learned this when you talked about the hormones, the insulin and the glucagon. So when blood glucose is low, the liver can break down that stored glycogen to glucose and then release that glucose into the bloodstream. The liver can also convert certain amino acids and lactic acid into glucose, and it can convert other sugars like fructose and galactose into glucose. So this raises blood sugar. But when your blood sugar level is high, like right after you eat a meal, the liver is going to convert that excess glucose into glycogen and store it so that it can be broken down, like we just mentioned, when blood sugar level is low. The hepatocytes store some triglycerides, break down fatty acids that we can use to make ATP, make lipoproteins, synthesize cholesterol, and use cholesterol to make bile salts. The hepatocytes also remove the amino group from amino acids so that they can be used for ATP production and converted into carbs or fats. Hepatocytes synthesize most of our plasma proteins such as albumin and prothrombin and fibrinogen. Remember, those are some of our clotting factors. The liver detoxifies substances like alcohol and drugs, penicillin, erythromycin, sulfonamides, into the bile. It can also chemically alter or excrete thyroid hormones and steroid hormones like estrogens and aldosterone. We talked about how the bilirubin is excreted once it's broken down. Obviously, the liver produces the bile salts. We mentioned that a few minutes ago. The liver can also store certain vitamins, A, B12, D, E, K, and some minerals like iron and copper, which can be released from the liver when the body needs it. The Kupffer cells of the liver phagocytize aged red blood cells, white blood cells, some bacteria, and the liver, along with the skin and the kidneys, is responsible for making the active form of vitamin D. Homeostatic imbalances of the liver include cirrhosis and hepatitis. Distorted or scarred liver as a result of chronic inflammation due to hepatitis, chemicals that destroy hepatocytes, parasites that might infect the liver, or alcohol are common causes of cirrhosis. So the hepatocytes are replaced by fibrous or adipose connective tissue. Symptoms of cirrhosis typically include jaundice, edema in the legs, uncontrolled bleeding, and increased sensitivity to drugs since the liver is unable to detoxify those as it passes through. Hepatitis is an inflammation of the liver that can be caused by viruses, drugs, chemicals, including alcohol. There are several types of viral hepatitis. Hep A is sometimes known as infectious hepatitis, and it's spread through fecal contamination, such as through clothes or food, toys, eating utensils. It's usually a mild disease in children and young adults. It can cause loss of appetite, overall just discomfort, nausea, diarrhea, fever, and chills. Eventually, jaundice usually appears. This type of hepatitis doesn't cause lasting liver damage, and most people tend to recover in about 4 to 6 weeks, but a vaccine is available. Hep E is caused by the hepatitis E virus. So you can see here that these different types are simply named based on the version of the virus that causes them. Hep E is spread just like Hep A. Although it doesn't cause chronic liver damage, Hep E usually has a very high mortality rate among pregnant women. Hep B is caused by the hepatitis B virus. It's spread primarily through sexual contact or contaminated syringes and transfusion equipment. It can also be spread through saliva and tears. It can produce cirrhosis and possible cancer of the liver. People that have the active Hep B virus can become carriers, and a vaccine is available. Hep C is caused by the hepatitis C virus, and it's clinically similar to the Hep B. It can cause cirrhosis and possible liver cancer. Donated blood in developed countries such as ours is usually screened for the presence of Hep B and Hep C. Hep D is transmitted like Hep B. A person actually must have been co-infected with B before they can contract D. Hep D results in severe limbage damage and has a higher fatality rate than infection with virus B by itself. But there is a vaccine available. The non-viral causes of hepatitis can be caused by drug toxicity and wild mushroom poisoning. If the bile doesn't have enough bile salts or excessive cholesterol, the cholesterol can crystallize to form gallstones. As they grow in number and size, they can lead to obstruction of the flow of bile from the gallbladder into the duodenum. Treatment can consist of gallstone dissolving drugs or shockwave therapy, which breaks them down, or surgery to remove them. For people with a history of gallstones that drugs and the shockwave therapy didn't work, a cholecystectomy, I never can say that word, removing the gallbladder and all of its contents may be necessary. More than 500,000 of these surgeries are performed each year in the United States. To prevent the side effects resulting from the loss of the gallbladder, patients usually have to change their lifestyle and their diet, including the following. Limit the intake of saturated fats. Remember, bile emulsifies fats. Avoiding the consumption of alcoholic beverages. Eating smaller amounts of food during a meal and perhaps eating 5 or 6 small meals a day instead of 2 or 3 large ones. And also it can be helpful to take vitamins and mineral supplements. Jaundice is when a yellowish coloration of the sclera of the eyes, the skin, and the mucous membranes results due to a buildup of a yellow compound called bilirubin. After bilirubin is formed, as we break down those red blood cells, it's transported to the liver where it's processed and eventually excreted into the bile. The 3 main categories of jaundice include prehepatic jaundice, which is due to excessive production of bilirubin. Hepatic jaundice, due to a congenital liver disease, cirrhosis of the liver, or hepatitis. Extrahepatic, outside the liver, is due to the blockage of the bile drainage by gallstones or cancer of the bowels or of the pancreas. Because the liver of a newborn doesn't function well for the first week or so, many babies will experience a very mild form of jaundice called neonatal jaundice, and that usually disappears as the liver matures. However, it's commonly treated by exposing the infant to blue light, and that converts the bilirubin into substances that the kidneys can secrete and get it out of the body.